Thank you, Natalie, and thank you to the ASBMB for giving me this award. It's a particular honor because, as Natalie was saying, Mildred is a point of reference for women in science, and in particular in biochemistry and biophysics, and it's a great privilege to receive an award in her name. So today I want to talk about work on transcription pre-initiation complexes, and I will show you in a second what the challenges are concerning such large and dynamic complexes. It makes it basically so far have been impossible. These lights are so bright, it's unbelievable. Okay, I'm gonna pass out. It really makes it very hard to study by more traditional structural biology, well-established methods like X-ray crystallography. So the methodology that we're using is single particle cryo-electron microscopy, and some of you may have heard about this, some may not be aware, so I'm going to do a one-slide summary of what is behind Cryo-EM. So just to tell you why it is that we use it, most important thing is we overcome the bottleneck of crystallization. The method is applicable to very large complexes. We study these large pre-initiation complexes. We also study microtubules. There is no problem with size. In fact, the larger the better for Cryo-EM. But most important for our transcription studies is that it requires very small amounts of sample. The kind of work that I will be telling you about, when we do our purification of these complexes on DNA, we end up typically getting 5 microliters at 50 nanomolar concentration. And that is the amount that that we use for the kind of work that I'm going to be showing you. So this, what allows us is to study fully assembled complexes that are very near physiological condition, and that means it's near because it's out of the cell, but otherwise it's under conditions where if you add nucleotides, the polymerase will start transcribing. The microtubules that we study will be assembling and disassembling. And because we don't have to recrystallize, we can study different functional states and have a very quick turnover of structures. And this is the case even when it is impossible for us to biochemically separate these states and they are mixed in the test tube. We can still computationally separate them in some cases and obtain structures in parallel. So what does this entail? We do have to purify our complexes. There's no sloppiness concerning the biochemistry. The cleaner your complex, the better you will do. And then the sample is placed on an EM grid. A few microliters, I was telling you, is blotted to a thin ledger, plunge into liquid nitrogen very quickly so that the water doesn't crystallize, to form this vitreous amorphous layer of buffer around your protein, and this then is kept at liquid nitrogen temperature, inserted in a transmission electron microscope. And of course, what we want to obtain is the three-dimensional structure of the object. Some of you may recognize what this is. And what we obtain in transmission electron microscopy is a two-dimensional projection of the object. And things are never this good because we have to fight the radiation damage that electrons can cause in a sample, so we have to use very low doses, typically of the order of 20 or 30 electrons per Armstrong square in the sample. So our images look more like this. They are very noisy. So to get from two-dimensional noisy projection to three-dimensional sharp reconstructions, we need to carry out two tasks computationally that can be done either one after the other or simultaneously. One involves identifying images of the same object in the same orientation, aligning them in plane, and averaging to recover signal. But the other is to combine, obviously, many different images from many different orientations at the right relative angles to go through the process of three-dimensional reconstruction. And this is what the process entails, and I'm not going to talk more about Cryo-EM. It's up to Jorgo if he wants to talk more about it. I'm going to move right into the system that we're studying. So this is the pre-initiation complex. It's a large macromolecular assembly that has to form at the beginning of every eukaryotic gene. And it is required because the polymerase, which is a very large enzyme, 12 proteins, 500,000 Dalton, itself cannot do two essential tasks. One, find the start for transcription, the beginning of the gene, and the other one, it cannot open the very stable duplex of DNA. So for that, it requires all this cohort of general transcription factors that you may have heard about from Bob Rader the other day. He was a major contributor to identifying this whole machinery. So TF2D, which is 1 million Dalton in size, is involved in recognizing core promoter sequences around the start site, and by doing so, then act as a nucleator for the assembly of the rest of the pre-initiation complex. TF2H itself, 10 subunits and 500,000 Dalton in size, is required, its ATPase activity is required to open the duplex. And then the rest of the factors are involved in help these two processes, and in many cases are involved in stabilizing the transcription bubble that forms just preceding the start of transcription. So these three larger complexes, the polymerase 2D and 2H, can yet not be reconstituted in heterologous expression systems, so we purify them in our case from HeLa cells. We study the human system. By immunopurification, we get tiny little amounts. And then we have to put them together, as well as these other smaller factors that are either monomers, dimers, that we can express in larger quantities in bacteria. We have to put them together on the DNA and purify it as homogeneous a complex as possible. So this is very challenging. And the only reason, and one of the things that we did was to simplify our life. So this is a disclosure that what we did was substitute all of TF2D, that 1 million Daltons, by just TBP, the Tata-binding protein, and to work using a core promoter that was Tata-based. Okay? And then to get this homogeneous state of a PIC, Yuan He, who was an exceptional postdoc in the lab that now has his own research lab at Northwestern, devised a methodology by which he used core promoter that was biotinylated, and he could bind to streptavidin-coated magnetic beads. Then we could add the factors to the DNA up to a certain stage in the assembly process, incubate with the DNA, and then wash off the excess and release the DNA-bound complexes by cleaving using a restriction enzyme. And one of the things that we did in order to make sure that we could interpret every region of the 3D map that we were going to ultimately generate was to build this complex one step at a time. So you'll see what I mean. So we started with the smallest complex that will contain the polymerase and will be bound to DNA. And this is a complex with TBP, TF2A, TF2B, and the polymerase itself. And the good thing about this is that there are crystal structures for all these elements. In fact, there are crystal structures of pairs, Pol2 with 2B, 2B with TBP, TBP with 2A, TBP with the DNA. So we would get the structure that we obtain have to be explainable with all those crystal structures, and that was the case. We could explain everything. The one thing that was missing is that we could only see the DNA, this region of the DNA, the part that is being bound in a DNA-specific, in a sequence-specific fashion by TBP and 2B. The rest was too flexible, and it was being averaged out. It meant that it was not being contacted by protein. So then we repeated this experiment, but we added, in addition to these factors, TF2F. And the idea is that now we'll see extra density that we will know has to correspond to TF2F. And in addition to, so for TF2F, there are crystal structures of 2 pieces, and we could dock these separately into the density and explain that density. I'll go into more detail on that in a moment. But one important thing is that this caused a conformational change in the polymerase where this clamp head region moves up, and now it engages the duplex DNA that becomes perfectly stabilized, and we can see now our whole construct. Then we added TF2E, and again, the new density had to now correspond to TF2E. There are no crystal structure of TF2E. There were some models based on cross-linking data, and they kind of qualitatively fitted. This actually topologically traps the DNA on the cleft of the polymerase, and then we added TF2H, this large 450 kilodalton complex, and And it went down here where we could dock the crystal structure of one of the ATPases, XPB, and see that it's contacting the DNA downstream of the start site, which is in here marked by the pink, corresponding to the INR sequence. So this we published some time ago, and in fact, this structure was not in that paper. What we had then was a negative stain structure, because putting together all of these was already hard enough. So eventually, we got the structure by Cryo-EM so that we could see the DNA, because otherwise it's invisible in negative stain. So we should have been happy, but we were not, because this structure was obtained with a direct detector. So those of you that are in the know of what is going on in Cryo-EM, new detector technology has really revolutionized the throughput, but also, most importantly, the resolution of the structures that we can obtain. And this structure was the same as this one, so this was very disappointing. But this was the problem. What I'm showing you here is now the same map, but it's colored by local resolution and filtered by local resolution. And I hope that you can see that this region, which corresponds to TF2H, is at much lower resolution than the rest. And this is an indication that TF2H is somehow moving. And we could sort out the data into two major states and see that what is happening. So if you take this kind of canonical view and you turn it around, so the DNA now is going like this, TF2H is actually moving, is moving around a central position and moving the DNA with it. It's just a slight motion, but it's enough to blur the density. So what we do in that case, this is something that I'm going to use a lot, so I'm going to concentrate a little bit on it, is that we go to a certain point in the refinement process, which is a process by which the experimental image is assigned a certain relative angle by comparison with a running a structure that we are reconstructing. When we get to this point, what we do is we mask out the part that is moving, and we adjust very lightly the alignment to better fit the model for just this part. And by doing that, you get better resolution for this region because it's not being blurred by trying to fit simultaneously this part. And by doing this, we got a significant improvement in resolution. And just for you to compare, this is where we were 3 years ago, where we were collecting data on CCD, and this was the kind of resolution regime that we had in the complex. And this is what we have right now. So the resolution has improved to the point that the secondary structure now is so clear that we can fit the crystal structures with very, very high accuracy. But most importantly, we can start to see things that could never be seen by crystallography. So I told you that TF2F, there were two crystal structures. One was of this domain, within RAP30, and this is the dimerization domain between RAP30 and RAP74. And connecting the two, there is a linker that had been shown to be important for the transcription process that, of course, crystallographers could not deal with, so that's why they cut it and crystallized the two pieces separately. Now we can see that linker starting at the end of one structure, moving around and connecting to the other, because it becomes stabilized in the context of the full complex. Okay, so this is the structure of what is called the closed complex, in which the polymerase is engaged with a duplex DNA. And this is the full complex, and this is what I would call the core PIC, which is without TF2H, where we are able to push the resolution. So in addition, we obtain an open structure by utilizing a mismatch bubble to mimic a transcription bubble. That structure for the core could go to significantly higher resolution. And then we obtain one of an initially transcribing complex that now includes a few ribonucleotides, so a little piece of RNA. And we have done these three structures in the presence of TF2S, which is an elongation factor, but also has been recently shown to be able to perhaps play a role in transcription initiation. But we knew that 2S can accelerate the process, the weak nuclease activity of the polymerase, to what we decided to also obtain one structure if the absence of TF2S. And this was good because actually this the RNA had disappeared from this structure. So we have 4 independent structures, and in particular, for the core complexes, they are of enough resolution that we've been able to generate now atomic models for these four structures that now we can compare to see what changes occur during the process of transcription initiation. So these are just two structures of the initially transcribing complexes, one with TF2S, the other without TF2S. This is just to show you, to give you an idea of the accuracy of the models for the lower resolution region like this. What we're doing is docking homology models as rigid bodies. And this is for the core of the polymerase where the resolution is the highest. So this is a set of beta sheets in different orientations. I hope you can see that we see beautiful separation and density for side chains. This is the active site where this is the bridge helix. In blue is the transcribed strand of the DNA. In red is the RNA. This is the fourth loop 2, and this is the non-transcribed strand. So this is the kind of resolution that actually are being obtained for crystal structures of the yeast polymerase. So for the polymerase itself, we are already at that kind of resolution, but we're working with the human system, and this came out of the full complex. So these are just some examples. This is TF2B, which plays an important role based on our structure on stabilizing that transcription bubble by making major contacts with the non-transcribed strand. And we compare this with the previous crystal structure of yeast that have a fragment of TF2B and was lacking the non-transcribed strand. And I hope you can see in green that for this region, the structures actually differ, and we don't know whether it is due to being yeast. More likely is due to the construct that was being used and being out of the context of a proper transcription initiation bubble. And this is what I was telling you about before. This is the wing helix of RAP30. This is the dimerization domain of TF2F. And this is that linker region. What you see behind are all of the other components of the PIC that TF2F interacts with, including many that are being contacted by this linker that becomes stabilized during the process of PIC formation. And those models, we have great redundancy because, remember, we have 4 different structures. This is the trigger loop for the aficionados. So some of them do not change during these 4 states, others change a lot, like TF2B. So I'm going to concentrate on that one for a second. This is just the region of the transcription bubble, but shown at those 4 stages. And this is just to tell you that before the bubble is open, most of this region of TF2B, the reader, the 2B linker, are not visible. And again, every time something is not visible, it means that it's flopping around so much it gets averaged out. But it becomes stabilized via interaction with certain elements of the polymerase that are indicated here and the transcription bubble itself. And then there is very little changes that occur from them on. This is now a reverse view. In that view, was only showing the density for the protein. Here is one, for simplicity, just showing you the density for the DNA, and this is the open complex. So you've seen the transcription bubble and the elements that bind to it. And I just want to show you what happens once you move through the transcription initiation process. Now you have 6 pairs of DNA, RNA. This is the upstream region that is being bound up here by TBP and TF2B. It cannot move. So the polymerase has to have been inserting the DNA, the downstream DNA, into the cleft. And you can see that the transcribed strand and the RNA are very nicely accommodated. But there is part of the DNA in the non-transcribed strand that is invisible. This is a region that is actually sticking out of the polymerase, exposed to solvent. And we think this is just a comparison between the two. This is a region of the DNA that is undergoing the famous scrunching that has been previously predicted or visualized, if you want, by biochemical or biophysical assays. Okay, so I completely abandoned TF2H because it was problematic, but we can do something about it. So we have 3 data sets that correspond to some kind of open complex, either open or early transcribing. So we pull them together, and we got a slightly improved density map, but this now has a lot of data. And we do the process that we did before, but in reverse. Now we mask out this part, and we concentrate on aligning the TF2H region. And again, we don't get quite as far, but we can improve the resolution, and this has allowed us now to generate a model based on fitting crystal structures or homology models of the different components. This is the DNA, which is beautifully visualized, the major and minor groups, the the XPB ATPase that acts as a translocase to push on the DNA. This is the other ATPase, SPD, and the rest of the components. So this is the first, the most detailed structure that exists of TF2H to date. Still a long way to go. But in this case, the most important thing is to look at the comparison between the closed and the open state. This is the position of XPB that I was telling you about, and our idea that fits very well with previously proposed models of it acting as a translocase is that it's placed in the right position to thread DNA into the active site of the polymerase. This is a superposition of the DNA in both states, where this place where they coincide, that they overlap, is right the position where TF2H engages. And we made a little movie. The protein is washed out, unfortunately, but at least the DNA you can see. This is where XPB is attached to the polymerase. This is the kind of motion that we can see for any given state. But if we now extrapolate from one to the other, XPB just stays here, pushes the DNA, and it is placed in the right position through the transcription initiation process to facilitate that. This is going backwards and forwards. Again, this is just an interpolation in chimera between the different states. Okay, so just very quickly, again, we've forgotten about TF2D because really this is the most difficult of these components. And this is due to the fact that, and this is work that Mike Cianfrocco did in the lab when he was a graduate student. He's now a postdoc at UCSD, so maybe he's in the audience, who knows, where he saw that TF2D, remember, is 1 million Dalton in size, and 1/3 of it, which we refer to as lobe A, actually moves within the complex by over 100 angstrom. And he was a biophysics student, so he liked quantitation. He looked at the position of that lobe with respect to what we call the core BC and showed this kind of bilobal distribution. And the thing that got us really very excited is when you give DNA and TF2A to the complex, this distribution shifts now to what we call the rearranged state. And when we do the EM, it ends up that only the rearranged state engages the DNA. So this was very exciting, and they told us something about the regulation, how TF2D, which has to actually interact with many activators, cofactors to regulate its affinity for DNA, has that inherent flexibility that now can be used by these factors to play with which state is ultimately present. But it's still very frustrating because the resolution here is really bad. This is what we call blobology. This is 30 angstrom. This is embarrassing. But just realize, look at the breadth of this distribution. This is not one state. This is many. So Robert, which is a present biophysics graduate student in the lab, set up to improve things a little bit. So the first thing is he used Yuan's method to be able to purify just the DNA-engaged TF2D. So we at least got rid of what we call the canonical state. Then he did cross-linking, used this direct electron detector, and the same trick for masking flexible elements. So he got this structure, which was already an improvement, and just for reference, this is what we had before. This structure we don't see anymore. But in here, what we can see is that of this yellow part, this region that is engaged with the DNA is very clearly visible. The rest is still so flexible that it's kind of smeared out. So what do? We mask it, and then we align in the rest, and this allowed us to improve the resolution sufficiently that we can take this crystal structure of TBP2A and the TATA box and fit it very nicely on this end of the DNA. Mike had already proven that this was the TATA end by actually labeling that end of the DNA with gold. And once you have positioned the TATA, the rest of the core promoter elements that we have used in this study were positioned within our reconstruction, and we could see that they were being touched by protein. But we didn't know what within TF2D was touching it. Now, this structure, again, when you look at local resolution, has a region that is better defined, and that corresponds to what we call the C lobe. What did we do? Mask that C lobe and do that one separately. And that, again, allowed us to improve the resolution a little bit more. And then we were very lucky because the lab of Zheng had obtained a crystal structure of human TAF1 bound to TAF7, and we could take that structure as a rigid body and fit it very nicely here, making contact with the MTE and DPE regions of the DNA. And they had predicted, based on the fact that they saw this beta wing that is very common in transcription factors, that it would be touching the DNA. And it was an amazing prediction. They had mutated these conserved residues and seen that they were important. And now we can see how not only these ones, but other adjacent residues are also involved in that contact. But something that was missing for their structure, and that has to do with this region here interacting with the INR, was a region that is missing that goes from here. So there's a break here in the structure that then restarts here. And that region, if you look at it, is actually quite conserved. And if you do a structure prediction, it predicts a very nice small globular domain that fits beautifully within the density. That means that we had identified major binders in TF2D of the core promoter. And the second was also, the last one was actually, again, very lucky, because some bioinformaticists found that TAF2, TAFs, by the way, stand for TBP-associated factors, I didn't do a very good introduction, is a homologue of aminopeptidases. And we could take one such a structure and fit it there very beautifully. And if you look at sequence conservation, a region here that is not conserved among the aminopeptidases, but is totally conserved among TAF2s, is involved in making contact with this MTE region. And this is now turning this around by 90 degrees and displaying both TAF1, 7, and TAF2 to show you that they bind on different phases of the DNA and that they interact very nicely with each other using residues that now we have identified. So I'm just going to end here. I'm going to summarize that we were able to dock this crystal structure and identify the position of the rest of the core promoter sequences. And then by zooming into this region, I haven't had a chance to explain how we identified these components also, but we were able to identify all the regions in TF2D that are involved in making sequence-specific contacts with different core promoters. And now if we combine both studies, because TBP2A and the DNA are in common, we can produce this synthetic structure of the TF2D base PIC that is really very telling. There is a beautiful complementarity in shape, except for a couple of very interesting overlaps that actually had been predicted to occur during the process of recruiting the polymerase, and that fit very well with biochemical data. But now we are up to over 2 million Dalton, where the resolution that we have, this is not filtered to the final resolution for all the components, but they go from 3.5 at the core active site of the polymerase to about 12 angstrom from the more flexible regions in TF2H. So just to thank again the people that did the work, our collaborators and our funding sources, and thank you very much for your attention.